ਪੌਦੇ ਦੇ ਸ਼ੂਟ ਸਿਸਟਮ ਵਿੱਚ ਪੱਤੇ ਅਤੇ ਤਣੇ ਆਉਂਦੇ ਹਨ। ਤਣਾ ਪੌਦੇ ਦਾ ਉਹ ਹਿੱਸਾ ਹੁੰਦਾ ਹੈ ਜੋ ਜੜ੍ਹਾਂ ਦੀ ਉਲਟ ਦਿਸ਼ਾ ਵਿੱਚ ਵੱਧਦਾ ਹੈ, ਰੋਸ਼ਨੀ ਦੀ ਤਲਾਸ਼ ਕਰਦਾ ਹੈ ਅਤੇ ਹਵਾ ਵਿੱਚ ਰਹਿੰਦਾ ਹੈ। ਫੁੱਲਾਂ ਵਾਲੇ ਸਾਰੇ ਪੌਦਿਆਂ ਦੇ ਤਣੇ ਹੁੰਦੇ ਹਨ। ਪੌਦੇ ਦੇ ਤਣੇ ਦੇ ਦੋ ਮੁੱਖ ਕੰਮ ਹੁੰਦੇ ਹਨ। ਇਹ ਪੌਦੇ ਦੀਆਂ ਜੜ੍ਹਾਂ ਅਤੇ ਪੱਤਿਆਂ ਦਰਮਿਆਨ ਪਦਾਰਥਾਂ ਨੂੰ ਲਿਜਾਣ ਦਾ ਕੰਮ ਕਰਦਾ ਹੈ। ਇਹ ਪੌਦੇ ਨੂੰ ਸਹਾਰਾ ਵੀ ਦਿੰਦੇ ਹਨ ਅਤੇ ਪੱਤਿਆਂ ਨੂੰ ਸੂਰਜ ਵੱਲ ਕਰਕੇ ਰੱਖਦੇ ਹਨ। ਇਸ ਦੇ ਇਲਾਵਾ ਕੁਝ ਤਣੇ ਪੌਦੇ ਲਈ ਭੰਡਾਰਨ ਦਾ ਵੀ ਕੰਮ ਕਰਦੇ ਹਨ। There is a tremendous variation in stem types and stem anatomy, although there are some common features which we will discuss. Today, our discussion about stems will include the study of internal anatomy and features of primary and secondary growth in stems.
Hello students, I welcome you all to today's lesson about the stem. ਤਣਾ ਪੌਦੇ ਦਾ ਉਹ ਹਿੱਸਾ ਹੈ ਜਿੱਥੋਂ ਸ਼ੂਟਸ ਅਤੇ ਬਰਡਸ ਪੈਦਾ ਹੁੰਦੀਆਂ ਹਨ। ਪਲਾਂਟ ਸਟੈਮ ਪੌਦੇ ਦੀ ਗਰੋਥ ਅਤੇ ਡਿਵੈਲਪਮੈਂਟ ਵਿੱਚ ਅਹਿਮ ਭੂਮਿਕਾ ਨਿਭਾਉਂਦਾ ਹੈ। ਇਹ ਪਲਾਂਟ ਦਾ ਸਟਰਕਚਰਲ ਪਾਰਟ ਹੁੰਦਾ ਹੈ ਅਤੇ ਇਹ ਲੀਵਸ, ਫਲਾਵਰਸ ਅਤੇ ਬਾਕੀ ਦੇ ਸਟਰਕਚਰ ਨੂੰ ਹੋਲਡ ਕਰਦਾ ਹੈ। The stems are more complex than roots in external structure, so it is not surprising that their internal anatomy is also very complex. Stem usually consists of three tissues: dermal tissue, ground tissue, and vascular tissue. The dermal tissue covers the outer surface of the stem and usually functions as waterproof layer, protect the inner tissues and control gas exchange. The ground tissue usually consists mainly of parenchyma cells and fills in around the vascular tissue. Its main function is to store food and conduction. It sometimes functions in photosynthesis. Vascular tissue provides long-distance transport and structural support. Most or all ground tissue may be lost in woody stems. The arrangement of the vascular tissues varies widely among plant species. Today we will look at variety of examples of stem anatomy which will help you to know more about the internal structure of stem. But before going into details, first let me share the learning objectives for today's lesson.
Upon completion of this topic, learners will be able to discuss about the primary structure of stem, describe structure of monocot stem, describe structure of primary dicot stem. List examples of monocot and dicot stems. Explain secondary growth in dicot stem. Discuss sapwood and heartwood.
Students, anatomy of a stem reveals a highly organized set of cell types. We can study the internal structure of stem by cutting very thin sections of stem and examining them under a microscope. So let us now study the internal structure of the stem.
Primary structure of stem. Transverse section of stem shows the following tissues: epidermis, cortex, endodermis, pericycle, vascular bundles, and pith. Let us discuss all these structures one by one. Epidermis. It is the outermost single layer of rectangular parenchymatous cells. It is covered with cuticle. Few stomata may be seen. Each stoma has two dumbbell-shaped guard cells. Epidermis may bear glandular or non-glandular, branched or unbranched hair of various shapes and size in dicot stem, whereas epidermal outgrowths are usually absent in monocots. Cortex. The cortex is distinct in dicots but not in monocots. It shows a great variation in plants. Usually, the outer few layers differentiate into collenchymatous, chlorenchymatous, or sclerenchymatous hypodermis. The remaining portion of cortex is made up of thin-walled, living, parenchymatous cells with intercellular spaces in dicot stems. Endodermis. The endodermis, if present, usually shows a layer of compact cells. The endodermis is the innermost layer of the cortex. The cells may or may not possess Casparian strips, but they contain abundant starch grains. This layer is also known as starch sheath. Pericycle. Pericycle forms the outer boundary of vascular cylinder. Pericycle may be single-layered or multi-layered. Vascular bundles. The vascular bundles are either arranged in single ring below the pericycle as in dicots or scattered in parenchymatous ground tissue as in monocots. Each vascular bundle may be conjoint and collateral, bicollateral, or concentric. The xylem is endark. The vascular bundles may be open, that is cambium present between the xylem and phloem, or closed, that is cambium absent. Pith. The pith is parenchymatous or in the form of hollow cavity in dicots or not distinct as in monocots.
Students, this was about the primary structure of stem. You saw the different tissues present in internal structure of stem such as epidermis, cortex, endodermis, pericycle, vascular system and pith. ਇਸ ਬਾਰੇ ਪੜ੍ਹਨ ਤੋਂ ਬਾਅਦ ਹੁਣ ਅਸੀਂ ਟਿਪੀਕਲ ਮੋਨੋਕॉट ਐਂਡ ਡਾਈਕॉट ਸਟੈਮ ਦੇ ਇੰਟਰਨਲ ਸਟਰਕਚਰ ਬਾਰੇ ਇੱਕ ਕਰਕੇ ਵਿਚਾਰ ਕਰਾਂਗੇ. So let us first discuss the internal structure of a monocot stem.
Internal structure of a monocot stem. The monocot stem shows only primary structure as there is no secondary growth in it. The common example of this type of stem is Z maize. The outline of the maize stem in transverse section is more or less circular. Internal structure of monocotyledonous stem shows tissues such as epidermis, hypodermis, ground tissue, vascular bundles. Let us discuss all these tissues one by one. Epidermis. It is the outermost layer of the stem. It is made up of single layer of tightly packed parenchymatous cells. Their outer walls are covered with thick cuticle. The continuity of this layer may be broken here and there by the presence of a few stomata. There are no epidermal outgrowths. Hypodermis. A few layers of sclerenchymatous cells lying below the epidermis constitute the hypodermis. This layer gives mechanical strength to the plant. It is interrupted here and there by chlorenchyma cells, which can synthesize food due to presence of chloroplasts in them. Ground tissue. It extends from below hypodermis up to the center of stem and is made up of thin-walled parenchymatous cells having intercellular spaces. They are smaller below hypodermis and become larger towards pith. The entire mass of parenchymatous cells lying inner to the hypodermis forms the ground tissue. Vascular bundles. Vascular bundles are scattered in the parenchymatous ground tissue. Each vascular bundle is surrounded by a sheath of sclerenchymatous fibers called bundle sheath. Each vascular bundle is made up of xylem and phloem. The vascular bundles are conjoint, collateral, endark, and closed. Phloem lies towards outside and xylem lies towards inside. It is endark, that is, protoxylem lies inside while metaxylem towards outside. The phloem in the monocot stem consists of sieve tubes and companion cells. It can be distinguished into an outer protophloem and an inner metaphloem.
So students, this was the internal structure of a monocot stem, and we studied this with the help of a Z maize stem. After studying the monocot stem, now let us study the internal structure of a typical dicot stem with the help of stem of a Helianthus anus, that is sunflower. So let us study this.
Internal structure of a dicot stem. A transverse section taken through the young stem of sunflower shows tissues such as epidermis, cortex, pericycle, vascular bundle, medullary rays, pith. Let us discuss all these tissues one by one. Epidermis. Epidermis is the outermost covering of the stem. It is represented by a single layer of compactly arranged, barrel-shaped parenchyma cells. Intercellular spaces are absent. The cells are slightly thick-walled. Epidermis shows the presence of numerous multicellular projections called trichomes. Externally, a thin, transparent, waxy covering called cuticle, which prevents excessive evaporation of water, surrounds the epidermis. Cortex. Cortex lies below the epidermis. the cortex is differentiated into 3 zones. Below the epidermis, there are a few layers of collenchyma cells. This zone is called hypodermis. It gives mechanical strength to the stem. Inner to the hypodermis, a few layers of chlorenchyma cells are present with conspicuous intercellular spaces. This region performs photosynthesis. The third zone is made up of parenchyma cells. These cells store food materials. The innermost layer of the cortex is called endodermis. Pericycle. Pericycle is the outermost covering of the stele, which lies immediately below the endodermis. It is represented by a few layers of compactly arranged sclerenchyma cells. Vascular bundles. The vascular bundles consist of xylem, phloem, and cambium. Xylem and phloem in the stem occur together and form the vascular bundles. These vascular bundles are wedge-shaped. Each vascular bundle is conjoint, collateral, open, and endarch. Phloem is of protophloem and metaphlloem types. Phloem consists of sieve tubes, companion cells, and phloem parenchyma. Xylem is of metaxylem and protoxylem types. Xylem consists of xylem parenchyma, xylem fibers, vessels, and tracheids. Medullary rays or pith rays. The primary medullary rays are parenchymatous areas between the two adjacent vascular bundles. The medullary rays connect the pith with pericycle and cortex. They provide pathways of radial conduction of food and water. Pith. The large central portion of the stem is called pith. It is composed of parenchyma cells with intercellular spaces. The pith is also known as medulla. Function of the pith is storage of food.
So ਵਿਦਿਆਰਥੀਓ ਇਹ ਸੀ ਡਾਈਕੋਟ ਸਟੈਮ ਦਾ ਇੰਟਰਨਲ ਸਟਰਕਚਰ ਜਿਵੇਂ ਕਿ ਸੂਰਜਮੁਖੀ। ਤੁਸੀਂ ਸਾਰਿਆਂ ਨੇ ਹੀ ਡਾਈਕੋਟ ਸਟੈਮ ਦੇ ਕੁਝ ਖਾਸ ਫੀਚਰਸ ਨੂੰ ਨੋਟ ਕੀਤਾ ਹੋਵੇਗਾ। ਡਾਈਕੋਟ ਸਟੈਮ ਦੇ ਕੁਝ ਡਾਇਗਨੋਸਟਿਕ ਫੀਚਰਸ ਇਸ ਤਰ੍ਹਾਂ ਹਨ। Characteristic features of a young dicot stem. Here are some of the characteristic features of a young dicot stem. Presence of stomata. Presence of a hypodermis made up of collenchyma. Presence of a wavy endodermis containing numerous starch grains. Presence of a bundle cap above each vascular bundle formed by sclerenchyma. Presence of fixed number of vascular bundles arranged in the form of a broken ring. Presence of conjoint, collateral and open vascular bundles with an endarched xylem. Now we will discuss the secondary growth in stem. Increase in girth or thickness of the axis due to the activity of lateral meristems, that is vascular cambium and cork cambium, is called secondary growth in thickness. Normally, it occurs in dicotyledonous stem. However, a different kind of secondary growth occurs in some monocotyledonous stem, such as in Dracena, Aloe, Yucca, Agave, Kingia, Sansevieria, Xanthorrhea and Lomandra, which is considered as abnormal or anomalous type. Basically, the secondary growth is responsible for growth in girth of the stem, produces secondary, that is new and more vascular tissues, provides a continuous connection of meristem cells between the primary tissues of the roots and the primary tissues of the shoots. Due to secondary growth in stem, some trees like bristlecone pines live to such a great age, that is several thousand years. So let us study the secondary growth in stem.
Secondary growth in dicotyledonous stem. Typical example of secondary growth shown here is the stem of sunflower. The process of secondary growth in dicotyledonous stem involves different stages. The first one is development of the vascular cambium. Secondary growth begins with the initiation of the vascular cambium, a cylinder of meristematic tissue that produces additional xylem and phloem tissues. Two regions of the primary stem contribute to the vascular cambium. These are fascicular cambium, which is the strip of meristem cells within the vascular bundle, and interfascicular cambium, the strip of meristem cells between the vascular bundles. In addition, towards the beginning of secondary growth, there is a process of dedifferentiation in some of the parenchyma cells of the medullary rays adjoining the vascular cambium. As a result, these cells now become meristematic and hence divide. The two strips of dividing cells unite to form a continuous ring of dividing cells, the vascular cambium, as you can see here. The meristematic cells of the vascular cambium are highly vacuolated and exist in two forms: fusiform initials, ray initials. Next stage is the activity of vascular cambium. The cells of cambium always remain in a continuous state of division. They mostly divide by periclinal divisions. Periclinal divisions result in the formation of two cells, one in front or behind the other. The mitotic activity on the inner surface results in the formation of cells, which differentiate into a set of xylem. It represents the secondary xylem. Similarly, The mitotic activity on the outer surface results in the formation of cells, which differentiates into a set of phloem. It represents the secondary phloem. Next stage is the formation of secondary medullary rays. The ray initials of vascular cambium ring divide by tangential divisions and add new cells. These cells remain meristematic for some time and then differentiate into parenchymatous cells of rays. These rays, produced by vascular cambium in between the secondary xylem and secondary phloem, are called secondary medullary rays.
ਵਿਦਿਆਰਥੀਓ, ਮੈਡੂਲਰੀ ਰੇਜ਼ ਦੇ ਬਣਨ ਤੋਂ ਬਾਅਦ ਐਨੂਅਲ ਰਿੰਗਸ ਬਣਦੇ ਹਨ। ਕੈਂਬੀਅਲ ਰਿੰਗ ਦੀ ਮਾਈਟੋਟਿਕ ਐਕਟੀਵਿਟੀ ਪੂਰੀ ਤਰ੍ਹਾਂ ਸੀਜ਼ਨਲ ਹੁੰਦੀ ਹੈ। ਇਹ ਸਾਲ ਵਿੱਚ 2 ਵਾਰ ਬਣਦੀਆਂ ਹਨ। ਇੱਕ ਵਾਰ ਬਸੰਤ ਰੁੱਤ ਭਾਵ ਸਪ੍ਰਿੰਗ ਵਿੱਚ ਅਤੇ ਇੱਕ ਵਾਰ ਪਤਝੜ ਭਾਵ ਆਟਮ ਵਿੱਚ। ਇਸ ਤਰ੍ਹਾਂ ਹਰ ਸਾਲ ਸੈਕੰਡਰੀ ਜ਼ਾਈਲਮ ਅਤੇ ਸੈਕੰਡਰੀ ਫਲੋਏਮ ਦੇ 2-2 ਸੈੱਟ ਬਣਦੇ ਹਨ। ਹਰ ਸਾਲ ਕੈਂਬੀਅਲ ਰਿੰਗ ਦੀ ਮਾਈਟੋਟਿਕ ਡਿਵੀਜ਼ਨ ਸਪ੍ਰਿੰਗ ਸੀਜ਼ਨ ਵਿੱਚ ਸ਼ੁਰੂ ਹੁੰਦੀ ਹੈ। ਸੈਕੰਡਰੀ ਜ਼ਾਈਲਮ ਸਪ੍ਰਿੰਗ ਸੀਜ਼ਨ ਵਿੱਚ ਬਣਦੇ ਹਨ। ਇਸ ਲਈ ਇਸ ਨੂੰ ਸਪ੍ਰਿੰਗਵੁੱਡ ਜਾਂ अर्ली वुड ਦੇ ਨਾਂ ਨਾਲ ਵੀ ਜਾਣਿਆ ਜਾਂਦਾ ਹੈ। ਜਦੋਂ ਕਿ ਸੈਕੰਡਰੀ ਜ਼ਾਈਲਮ ਜੋ ਕਿ ਆਟਮ ਵਿੱਚ ਬਣਦੇ ਹਨ ਨੂੰ ਆਟਮ ਵੁੱਡ ਜਾਂ ਲੇਟ ਵੁੱਡ ਦੇ ਨਾਂ ਨਾਲ ਜਾਣਿਆ ਜਾਂਦਾ ਹੈ। The two distinct layers of secondary xylem, the inner springwood and the outer autumn wood, together represent annual ring. One such annual ring is added every year due to secondary growth. Thus, it is possible to count the age of a dicot tree by counting the number of annual rings. Thus, secondary growth in the steel results in the formation of secondary xylem and secondary phloem. Due to the addition of these tissues, there is an increase in the girth of the steel. As secondary growth continues to occur, several changes take place in the secondary xylem. In a much older stem, the secondary xylem or wood shows 2 regions, namely heartwood and sapwood. Let us check these types of woods.
Heartwood. Heartwood is also known as duramen. It represents the centrally located inactive portion of the secondary xylem. It is incapable of conducting water and provides only mechanical support to the trees. Sapwood. It is also known as alburinum. It represents the peripheral, active portion of the secondary xylem. This portion is still capable of conducting water. Due to its meager mechanical strength and lack of durability, the sapwood cannot serve as commercial wood. The next step in secondary growth of stem is formation of periderm. The mitotic activity on the inner surface of the cork cambium results in the formation of cells which undergo differentiation into a living tissue called secondary cortex or phyloderm just above the primary cortex. The mitotic activity on the outer surface results in the formation of cells which undergo differentiation into a dead tissue called cork or phloem, just below the epidermis. The cork covers and masks the hypodermis. The tissue resulting from secondary growth in the cortex, the cork cambium, and the secondary cortex together represent a region called periderm. The periderm, along with the primary cortex, represents the bark. In several dicot plants, the bark peels off regularly. Due to the formation of periderm, the epidermis is subjected to pressure, and as a result, it breaks at several places to form openings called lenticels. The lenticels, also known as aerating pores, enclose a group of living cells called complementary cells. Through these, cells exchange of respiratory gases and to some extent transpiration takes place. Thus, secondary growth in the cortex results in the formation of periderm. Due to the addition of this region, there is an increase in the girth of the cortex.
Students, the type of secondary growth which deviates from the normal type is called abnormal or anomalous secondary growth. It occurs either due to abnormal position of cambium or by abnormal activity of cambium, and it occurs in some monocot stems. So students, enough for today. Let us quickly check with some short questions what you have grabbed today. Be attentive, here are some questions. The first question is, what is the location of cambium in a dicot stem? The answer is, location of cambium in a dicot stem is intravascular between xylem and phloem within each vascular bundle. The next question is, what type of vascular bundle is found in dicot stem? And the answer is, the vascular bundles are conjoint, collateral and open vascular bundles with an endarched xylem. My next question is, which tissue is commonly called wood? The answer is, secondary xylem is commonly called as wood. The next question is, what is annual ring? The answer is, the two distinct layers of secondary xylem, the inner spring wood and the outer autumn wood, together represent the annual ring. Hope I have made a successful attempt to clear your concepts regarding the primary dicot stem, monocot stem and secondary growth in dicot stem, which will help you in fetching good marks in the exam. Thanks for your cooperation and attention. Looking forward to the next class. See you then.